All right, in this video, we're going to begin a review on Unit 3. Unit 3 covers three main topics. The first one is enzymes and energy. The second one is photosynthesis, and the third is cellular respiration. So let's begin by talking about this idea of enzymes and energy. Enzymes are chemicals that speed up chemical reactions, and they speed up their chemical reactions by lowering their activation energy. We're going to talk about that shortly. Enzymes are typically proteins, and when you're talking about the enzyme interactions, there's going to be a couple of words involved. Obviously, first you have the enzyme. Second, you have the substrate. The substrate is the reactant in this reaction, is the thing that is going to be changed from this reaction. It's important to note that the enzyme is unchanged in this reaction. The enzyme is like a tool. When you take a tool to a job site, you use that tool, you pack that tool away, you're able to use that tool again. It's the job site that's changed, not the tool. So in this case, it's the substrate that's going to be changed, not the enzyme. The enzyme has a place where the substrate fits. It's called the active site. You don't have it, I don't have it labeled in this picture, but this is the active site, and notice this enzyme fits this substrate particularly. Sometimes you'll see the word induced fit associated with that. Notice it doesn't look exactly like the substrate, but when you kind of bind it in there, it fits around it, very similar to the way that a latex glove fits on a hand. It's definitely designed for a hand, but it kind of molds around the hand. And a lot of times you'll see this term enzyme substrate complex associated with when they are bound together like this. This also continues to affirm the fact that the shape of a protein determines its function, and that's why the shape of a protein is very important. And so this particular interaction shows that definitely. Just real quick on the naming of enzymes, almost all the time you're going to see this ASE at the end of an enzyme, and the name will typically tell you what it's doing. So if you know these Latin roots, you can usually, pick it out. Like hydrolase is going to have something to do with hydrolysis, right? And we're not going to go over all these. You can get the idea. These enzymes basically tell you what they do. A lot of naming conventions for enzymes are pretty simple. Enzyme reactions are going to usually fall into one of two categories: anabolic reactions and catabolic reactions. Catabolic reactions have to do with breaking things down, taking something and breaking it into multiple pieces. An anabolic reaction is one that is going to build them up. The way that I remember this is I think about anabolic steroids, where the purpose of anabolic steroids is to build muscle. So it's anabolic, and then catabolic, I think of cats destroying things. Whatever works for you, do that. And then what does that have to do with activation energy? Well, activation energy is the amount of energy that's necessary to start a chemical reaction. And so this shows the standard kind of reaction progress and different kinds of reactions here. In this particular reaction, you have what is called an exergonic reaction or a catabolic reaction. Actually, both of these are those. And how do you know? Well, you see here there's a certain, so you have energy over here on the y-axis. There's a certain amount of energy in the reactants, and as the reaction progresses, most of that energy is going to be released. This EA is the activation energy. This little hump represents the amount of energy that's necessary to get that reaction started. And this purple line represents the time that the reaction takes to occur. Same here. This one is also a catabolic reaction or an exergonic reaction. Sometimes you'll hear them called combustion reactions, kind of an older term. Notice a lot less energy is released here, but a lot more energy is necessary to get this reaction started. So what do enzymes do? Well, here's a picture of that. Notice the enzyme here, this is activation energy necessary for the enzyme. So it lowers that activation energy. And what does this do? In effect, it's going to speed up the progress of the reaction. The progress for this reaction has to go all the way up here and then come all the way down. This line is much shorter. It's just a simple way of explaining it. But then the energy that is necessary to start this reaction is lowered because of the presence of the enzyme. How do you think about this in just real simple terms? If the way that I describe it to my students is I explain to them a situation where I would put all of my students out on a football field in random locations. And if I ask them to close their eyes and spin around randomly, how long is it going to take it before they join hands with another student and then stop moving? It's going to take a long time. But what if I assigned two of my students and said, you are the enzymes and your purpose is to go and grab two hands and put them together? It's going to speed that up significantly. And so the enzyme provides a place for reaction to occur, and it's going to speed that reaction up, thus conserving energy and speeding up chemical reactions so that living systems can do their thing, which is live. Here's an example of an anabolic reaction. How do you know? Well, here I have the reactions, energy on my y-axis, reaction path, the reaction progress on the x-axis, and I start with a certain amount of energy. This amount of energy that is necessary to start the reaction again is called the activation energy, and notice at the end I have more energy available than I had at the beginning. So we will typically call this an anabolic reaction, or an endergonic reaction is another word that you'll hear for that kind of reaction. And that brings us to this idea of energy that's located in living systems. We're going to talk about two reactions, photosynthesis and cellular respiration. Photosynthesis is an endergonic reaction in that it creates a product that has more energy than what was at the beginning, and then cellular respiration is the opposite of that. It's going to take that product, break it down to get energy back out of that product. The sun is going to be the source of energy, and then ATP, or chemical energy here, is going to be the output of that energy. These two reactions are coupled. We're going to talk about that too. This brings us to these two types of organisms, autotrophs and heterotrophs. Troph is just feeding of some kind. Hetero means other things, feeding on other things, and auto means self-feeding. And so it's just two types of organisms, 8th grade science there. And the second law of thermodynamics. This is important when we talk about our reactions because first law of thermodynamics says energy is not created or destroyed. It's only transferred or transformed. It's changed or moved. And so the second law has this. Well, some energy is lost in those interactions. And that energy is lost as heat because everything tends toward disorder. Heat is a very disordered form of energy, and so when these reactions take place, some of that energy is going to be lost or some disorder is going to be created. There is no such thing as a completely ordered reaction to where we would have world peace, but we don't. So these reactions lose energy, therefore we lose resources, therefore we are constantly feeding into that. Even the sun itself is not an infinite source of energy because it will eventually dissipate into heat as well. And here are those reactions that we went over. Some other graphs to kind of show you those reactions. You start with the reactants, have less energy than the products. That is an endergonic reaction. A lot of this Gibbs free energy stuff that we don't need to talk about as much anymore. But anytime you see that free energy, it's talking about the amount of energy that's available to do chemical reactions. So what is happening over the course of an endergonic reaction is the amount of energy available is increasing, usually because of some energy output from the outside, like the sun. An exergonic reaction is the exact opposite. When you're finished with the reaction, you have less energy available. When you eat food and your body breaks that food down, getting the energy out of it, and spends that energy, there's less energy available at the end of that expenditure. It makes sense. And one of the reasons is because some of that energy is going to be given off as heat, going back to our second law of thermodynamics. Energy is also given off as heat here as well. And I think that's important to note, that all chemical reactions have some measure of entropy in them. And the Gibbs free energy equation shows that, but again, you're not going to be required to know that. And this brings us to energy coupling. Not sure why I have the Pokemon in there in the lower left corner, so you can ignore it. Energy coupling has to do with two reactions being necessarily paired together. Photosynthesis, cellular respiration are two great examples of this that every biology teacher is going to talk about. Here's another one talking about the creation of ATP. ATP is a molecule that represents the kind of the energy currency of the cells, and the reason it has energy is because it takes a lot of energy to shove that third phosphate onto the end of that molecule. Phosphates really don't like each other, and it's kind of like two holding opposite ends or same ends of a magnet together. There's a force that's holding them together, right? And so that ATP is kind of just spring-loaded, ready to go. Well, when hydrolysis of ATP takes place, remember hydrolysis, adding water in order to break things down, it's going to release that energy. But when you do, you have an ADP, which represents adenosine diphosphate. This is adenosine triphosphate, didn't mention that. And then you have this phosphate that is just kind of sticking out there. Well, in order to put that phosphate back on, it's going to require an input of energy, which is going to also spend or make a water, dehydration synthesis. And the reason that the battery is used, I think it's a great example because it's something that's near and dear to the heart of every student. It's their phone. A charged battery represents something that has energy, right? And so when you start to use the battery of your phone, those electrons are moving throughout the phone, and then they're powering the systems. Well, a battery that is dead basically represents there are no more stored electrons, or you could even say that the battery is at equilibrium. The electrons there's no net movement of electrons. And so in order to power that battery back up, you have to provide energy from the outside by plugging it into the wall, storing up a concentration gradient of the battery and making it alive again, so to speak. And this brings us to reaction pathways. Enzymes are going to oftentimes be used in pathways, and just as the simple idea of this is that you have A, enzyme 1 is going to make B. Well, B is not the finished product, right? B is just kind of an intermediary product, and enzyme 2 is going to make B into C with a second reaction. Enzyme 3 then is going to make C into D, and D is the final product. The reason this is important because photosynthesis and cellular respiration both represent pathways in which each step along the way is not the end of the process, but it represents kind of an intermediary process, and enzymes are involved all along the way. We don't talk about all those enzymes in this class because it would be extremely arduous to do so, but we just talk about pathways in general so that we understand that. And another concept that is important to AP biology is this idea of conformational shape. Conformational shape just has to do with the shape of an enzyme or the shape of the substrate, and there's a conformational shape change associated with that reaction. So when the enzyme binds with the substrate, you have a shape change, and when that reaction occurs, you have another shape change. Conformational shape change usually requires energy or will kind of take energy out of the system, like an energonic or exergonic reaction. And I have more stuff to talk about. Denaturation. Sorry, I don't have a list of my slides in front of me. Denaturation has to do with the change of a shape of a protein. And so why is protein shape important? Well, protein shape determines that protein's function. Without having a particular shape, it can't do its particular function. There are different things that will denature a protein. Remember, we talked about that most enzymes are proteins, and this enzyme requires this shape to do this function, right? And so if you take away the shape, you take away the function. There are several things that can cause a protein to denature. Let's talk about those. Temperature is one of them. All enzymes have what are called an optimal temperature. They like to operate at that temperature. You'd expect the enzymes in our body, for instance, to really like 98.6 degrees. Not all enzymes in our body require that temperature, though, because our body has varying temperatures depending on the part of the body, depending on the function of that part. But in general, 98.6 is the optimal temperature for the enzymes in our body. It's going to change from one body to the next. Bacteria that really love hot springs are going to want their optimum temperatures to be much higher, and they couldn't survive in our bodies, for instance. It would be too cold. And so temperature, this graph is going to show you that. You have slow, which you'd expect, right? Slow gets to optimal rate once it gets to optimal temperature. And then what happens here is that protein completely denatures. That's why there's zero activity after a certain point, because that protein will not do the job that it's been asked to do. Same with pH. There's going to be an optimal pH for enzymes. pH of human blood is 7.4. The enzymes in that blood want to be 7.4. But other places in the body don't have that. For instance, there's an enzyme in the stomach called pepsin. It loves this low pH. Take it outside of the stomach, it stops working. It has an optimal pH. It wants to be at that particular pH. pH causes proteins to denature for several reasons that are beyond the scope of this class. Substrate concentration is also going to affect the rate of enzyme reaction. Now, this is important. Here's the way I describe this particular phenomena. If I have 10 students in my class that I've asked to staple papers, and I have a stack of 10 papers for them to staple, then that reaction is going to take place at a certain rate, right? 10 students all have a paper to staple, and they're all going to be doing it. Well, if I only have 2 papers, then all 10 of my people are needed to do that. But what if I have 500 papers? I can only still do 10 at a time, right? And so what this represents is the amount of substrate that's available is going to increase as long as there's enzyme. If there stops being enzyme, then the rate can't increase anymore because they're kind of full. They can only operate at a particular rate based on the amount of enzyme that's available. And when that substrate disappears, what do you expect to happen? The rate's going to go down or disappear completely. All right, so increasing concentration does not affect reaction rate necessarily because there's just a certain amount of enzyme. That's what I mean by point of saturation. Can't be any more work being done. And then enzyme concentration, well, let's take that same stack of 500 papers, and let's say that I increase the number of students to 500. Well, then it's 1 for 1. And so by increasing the enzyme that's available, I directly increase the rate of reaction. So this shows that by increasing the enzyme concentration is directly proportional to the rate of reaction. This is the last slide. Inhibitors. There are certain things that can chemically alter an enzyme, and these are called inhibitors. The word inhibitor helps us with that. And there are two types of inhibitor, competitive inhibitors and non-competitive inhibitors. Really simple. Competitive inhibitors compete for the active site. So a competitive inhibitor will actually bind to the active site, keep it from working, physically block the substrate from binding to it. A non-competitive inhibitor is going to bind to what's called an allosteric site. Allosteric literally means other shape or other arrangement. And so there's another site that this non-competitor inhibitor binds to. Well, when that chemical binds to this chemical, this shape, it's going to cause the shape of that enzyme to change. If the shape changes, the function changes, the substrate's no longer able to bind. And so both of these are able to slow down rate of reaction. Competitive inhibitor is going to slow it much more because it directly competes for the active site. But both of these are going to have an effect on the rate of reaction. I haven't seen any graphs that are going to have you tell what's the difference between competitive and non-competitive inhibitors as far as the rate of reaction, but the actual content, understanding allosteric sites and active sites and the competition therein is important. Hopefully this was helpful. If this was helpful, why don't you like it? Why don't you subscribe to my videos? I post videos all the time, hopefully helping you to do well in AP Bio and beyond. If you feel like this, if you're going to continue your biology career, this would be a great review session for those 101 class, not really 101, but like, the first year biology classes that you take as a freshman. Very good. Lots of my own students come back and tell me, I didn't even have to take notes in class. I just used your notes, and me and my friends watched your videos. And so encourage you to continue to do so as well. Again, please subscribe, like this video. Hopefully this was helpful. Thank you for watching.